Yes, good evening to all the participants. So welcome to the 12th session on pre-cell epitope prediction. So in certificate and internship course on artificial intelligence and machine learning approaches for drug and vaccine design in cancer. Now, let me, before going to this today's session, can we recap the last session topics? So what are the last session topics we have discussed? Because, um, this is the one of the major chapter in vaccine design. Okay, if you see the schedule, okay, this is the schedule. In the schedule, you can able to see we have majorly target discovery, drug design, system biology, vaccine design, and the last two sessions will be one session will be in machine learning, and the other session will be in the artificial intelligence. Okay, so we have two more sessions. One is on tomorrow and day after tomorrow. That is on Monday. Okay. So your coordinator is going to communicate you with you to fix the date for MCQ. So I'll be completing my sessions on Monday. So tomorrow and day after tomorrow you have the session other than today's session. So we can just recap today's session contents. I mean last session contents. So very good. For the first time, I'm getting an answer from Sindhu that we have discussed something about IEDB. Can someone expand what is IEDB? What is the expansion of IEDB? So where exactly we have utilized this IEDB? Even the other participants can interact. Only few participants, those are interacting or interacting. I anticipate to everyone to interact if you understand the concepts. That it is immunoepitope database. Very simple expansion, isn't it? Yeah. So what else we did? We discussed. Yes, as usual, I got answers from Veera, Rahul that we have discussed something about MHC binding. And what else did we discuss? So what are the two types of major molecule MHC molecules is present in IEDB database? There are totally three types of MHC molecules, isn't it? But what are the two major MHC molecules you can find in this IADB? So this is my next question. A participant join on time. Now the time is 7:04. Join 5 minutes earlier. If you have missed the earlier sessions, you can't able to track up what's going on in today's session. So today is the last session for vaccine design. Then I have to go for artificial intelligence and machine learning approaches. Okay, now let me discuss about this. Related to vaccine design, any doubts, you can also clarify it on today. Yeah, what else? I just asked you a question, like what are the two major MHC molecules present in IEDB? There are so many kind of epitopes already deposited in IEDB, isn't it? So we have because many of them don't study immunology if they are in second or third year. Only in final year they may find this subject. So if you interact, even the others can learn from you. It looks like some simple question for the final year UG or even PG. But how about the others? Even others should also able to understand, isn't it? T cell epitope and B cell epitope. Good. But my question is, what are the two major MHC molecules? Kind of information is already present in IADB. One is MHC1, the other is 2, isn't it? Only these two molecules are present in the database. Whether they mention anything about MHC3? No. Then what are the different parameters of, yes, good. But now my next question is, what are the different parameters of MHC1 and 2 we can calculate in IEDB? There are plenty of tools in IEDB, isn't it? IEDB is not just a database. We also have plenty of tools. So what kind of parameters like processing parameters like that. Do you have any idea? We generally go for MHC. Good. Again, I got answer from Veera Rahul. Even the others can also interact. I suggest that everyone can watch the recorded videos if you can't able to attend the sessions on time. Okay. Yes, I also got answers from Nandini that something related to CD4 and CD8 we have discussed on the last session. And also we discussed something about epitope binding, that is epitope binding with the paratope of antibody, isn't it? Epitope generally binds with the paratope of an antibody. So related to binding, so we call this as not only we call this as a presentation, an antigen presentation. The MHC binding, we call it as an antigen presentation, and processing, we call it as an antigen processing. The antigen bind with MHC molecules can play two important functions. One is antigen presentation, that we call as a binding, and the other is the antigen processing. Okay. Okay. Now, if you see this immunogenicity, okay, the immunogenicity, like if any epitope, is considered as an immunogen, then we can check these parameters through the immunogenicity. And we have also seen what are the various parameters. We use it for calculation of immunogenicity. Okay. Here, if you check the immunogenicity of an epitope, it tells whether it's an immunogen or not. Yes or no, like that. Even there is one option called toxicity. Even you can predict the toxicity of an epitope. It tells just a toxic or non-toxic. That's it. Okay. So similarly, if you see this, yeah, we also discussed about binding, and then we also discussed various informations on epitope predictions. Good. So these are the informations we have discussed on yesterday, isn't it? So generally, you try to predict the epitopes from a viral antigens or cancer antigenic proteins. Whatever epitope you have predicted, if it is in cancer biology, you obviously work on cancer antigen proteins, and whatever epitopes you predict from the cancer antigenic proteins, you try to bind with the paratopic region of what kind of a paratopic region? MHC molecules. The MHC molecules, we check their binding, definitely. So how these peptides can be docked with proteins? So I told you we can use autodock. Autodock is the tool used for binding the peptides with the proteins. But how to model the 3D structure of peptides? Because today we'll be discussing about B-cell epitope prediction. There you should also you should have a knowledge on how to predict the 3D structure of peptides. Now I take you to the server PepFold3. So there is one server called PepFold3. Okay, so there are so many kind of softwares. There is one software called ACD Chem Sketch. Okay, even through the ACD Chem Sketch or Vega. So there are some software tools where you can also design the peptides. If you have a licensed version of Discovery Studio, there also you can able to make it up. But with the freewares, if you want to try, you can use this PepFold3. In Google, I typed like that. I got this information. Here you got something like run PepFold3. Simply click run PepFold3. If you click this, okay, earlier we have got some results for MHC1 by presentation as well as MHC2 presentation. You remember on yesterday, I fed my email ID to generate the results to my email ID. So I got the results in my email. Okay, so whatever I got, okay, so I just opening and showing it to you. So I already told you, if you get a results, you can also download the results in the Excel sheet, in the form of an Excel sheet. But whatever results they have sent here in the form of .csv, okay, just opening it. I told you we can take the best peptide based on the score. If you get a maximum score, that is the best peptide. For example, if this is the best peptide, okay, if this is the maximum score, and if this is the best peptide we have received, so you have to type that peptide. Okay, if you can download in the desktop and you can open it in the Excel sheet, okay, and then you can copy and paste it. We can check now first what is the base peptide based on the score. So what I'm going to do here is I keep here, okay, the score, and I'm going to keep something like a filter, or I just sort it out based on the ascending order. Okay, so this is the best peptide actually. When I sort, so I got this as the best peptide. Okay, so this is the best peptide. So now we have to simply copy this. We have to simply copy this peptide, and then we have to paste it in this pepfold3 server. Just like that, you have to get the best peptide, and if you want to get a 3D structure of it, open pepfold3 and then paste the peptide. Now check all these nearly 100 simulations we are running at a time, and we are sorting models, and we are getting if you want to get, simply click run. Okay, so these are the default parameters, no need to change anything. Simply click run. Okay, it's asking to type the captcha. I'm typing the captcha. Now you are going to get this. Okay, yeah, it takes some time. Okay, immediately won't get the result. Similarly, however, I tried for MSc1, even you will be getting for MSc2. Also, we got MSc2 results here. You know, the data I generated from the IEDB website, it's in the web resource itself. Okay, after pasting the W protein of Nipah virus, I'm getting the best epitopic peptides based on the scores. I'm taking this best peptide, okay, and I'm just copying it and pasting it in the pepfold3. You know, actually for the MSc1, we have provided 9 length epitopic peptide size. For MSA2, it is nearly 13, isn't it? So whatever size we fixed and whatever allele we have fixed, so we got the answers for that. I think presently we have designed uh, for an Andhra Brahmin people. Okay, so the Andhra Brahmin, I have selected that kind of an allele. And for the Andhra Brahmin, this kind of an epitopic peptide, whatever we have designed, can be used as a vaccine for treatment of Nipah virus infection. Similarly, you can try for even cancer protein. Okay, so this is some information. So if you get a result in pepfold3, I can show you. Meanwhile, I can take you to the presentation of today's session, that is on B cell epitope prediction. So before going to the B cell epitope prediction, I also give some fundamentals on adjuvants. Do you remember? I was telling that I will be providing you some information on adjuvants, then I go for B cell epitope prediction. Yeah, if you see the adjuvants, okay, I also discussed few important properties of adjuvants. Can you interact with me? What are the important properties of adjuvants? There are so many functions of adjuvants. There are so many functions of adjuvants. But what are the important properties or functions of adjuvants? Why we have to use adjuvants in the vaccines? If you see even the COVID-19 vaccines, many vaccines have adjuvants. What is the need? Why we have to think about this? Of course, this is an essential component nowadays to prepare the vaccine. Enhances the action of vaccine. Actually, action, you mean the efficacy, isn't it? How the efficacy of a vaccine construct or vaccine is improved by adjuvants. What are the properties? There are some immunological properties, isn't it? What are the immune properties we have discussed on the last session? So what are the immune properties? Why we have to go for, vaccine is nothing but a biological preparation to combat the foreign antigens entering into our body. Yeah, so to provide the antigenicity, we go for vaccines, of course, so to improve the antigenicity. Good. So number one, first property is to improve the antigenicity. We can use the adjuvants. And I was also, when I given an example like COVID-19 vaccines, I told it can be Covishield or Covaxin. You have to go for 2 boosters. Minimum, you have to go for 2 boosters to boost the efficacy. To increase the efficacy, you should not limit up to the first booster. Even if you are getting Covaxin after the second booster, I told you Covaxin after the second booster of Covaxin, you may get maximum of 91% efficacy in Indian population. If so, if you see the first booster, it comes around 50 or 60, only 60% efficacy. But only after giving the second dose, it increased up to 90 to 91%. That too for the earlier strain of SARS-CoV-2. We can't take it for the other 99 strains of SARS-CoV-2. We have alpha, beta, gamma, delta, iota. So many strains are there. Each and every strain you may also have 4 to 6 mutations. Okay, so for each and every mutation, of course, we have to design a unique kind of a vaccine. So even if you are designing a peptide, obviously you have to design a peptide of a unique thing according to that. Okay, so of course, I also told something about precision medicine and also precision vaccine. So if you attend the last session, I might have mentioned that if you want to prepare a vaccine for a specific Indian population, if you take in Andhra, if you want to take for Tamil Nadu or for a Delhi people, you have so many kind of analytics. So you select the analytics and you prepare accordingly. So okay, till now I didn't get any answers. Only one has interacted with me. That is related to only the antigenicity is improved. Other than antigenicity, what else did we discuss? I think many of them forgot. Then I have to take you to the earlier presentation. So in the reverse vaccinology and immunoinformatics, I presented this slide. Do you remember? So it not only makes the antigen more immunogenic, but to produce high antibodies. This is most important. Why I'm taking you to this is because in B cell epitope prediction, this is the desired property we can anticipate when compared to the T cell epitope. We'll be producing more antibodies in that B cell epitope. And we also generate more specific cytotoxic T cells and memory T cells. This is what we discussed in the last session. The last session I told you, not only the antigen presenting cells try to combat the cancer cells, but also whatever cells, T cells interact with the corresponding antigen presenting cells where it has an antigen on the MHC surfaces. Okay, it gets a kind of a memory. The T cells will get a kind of a memory, and the T cells act accordingly to combat the corresponding cancer antigens. I mean, cancer cells. Okay, by interacting with the cancer proteins. So this is how you can combat. Okay, so now we'll be discussing about also adjuvant and its properties. Actually, there are we have worked on several adjuvants. We have worked on several adjuvants in biotech labs in Sri Venkateswara College of Engineering. So the type of adjuvants what we worked, so especially in my PhD research is IL-2. I worked on IL-2, IL-4, interleukin-2, interleukin-4, granulocyte, macrophage colony stimulating factor. Of course, this is a kind of a growth factor. Okay, and IL-4 and IL-2 are meant for increasing the T cells. These activate the T cells. Of course, these interleukins are important. And if you see this AH03, AH01, so I worked on even AH03 adjuvant. And also the dendritic cell itself can act like an adjuvant. Dendritic cell itself can act like an adjuvant. I told you adjuvant has majorly 3 important functions, but there are plenty of functions with the adjuvants. Earlier in 1930s and 25, something like that, you got alum, aluminum salts, so especially alum, aluminum phosphate, so many kind of aluminum salts came into existence to increase the efficacy of vaccine. Later, we get a friends adjuvant, okay, and we have these kind of other kind of particles and other kind of biological molecules. So all these things we have used. But after 2010, that is in this recent decade, whatever I mentioned, okay, these are the major molecules published in the reputed journal papers indexed in PubMed. Okay, so even I worked in almost all these kinds of adjuvants in my lab. So in my lab, I worked on even plant-based adjuvants, where these are the plant compounds got from the Beribinum B got from Beribia diffusa. In Tamil, we call this plant as a Mukarate Kirai, and Ashwagandham, that what we call it as a Withanea somnifera. And here I got a vithanol compound. In these antigens, BCG, tuberculin, tetanus toxide. So I just worked on the effect of my plant compounds on the uh, dendritic cell life stages of dendritic cell development. If you see dendritic cells, initially the dendritic cells are originated from the monocytes. From monocytes, we will be getting immature dendritic cells, and from immature dendritic cells, we will be getting mature dendritic cells. Okay, so if you see this, these adjuvants can act on the monocytes and also act on the immature dendritic cells. Okay, if it acts on the monocytes, it creates the immature dendritic cell, and if it acts on immature dendritic cell, you also generate mature dendritic cells. Of course, you should need an antigens also for uh, maturation of mature dendritic cells from the immature dendritic cells. Okay, so this is a kind of a life cycle. And of course, this I worked in my PhD research. Not only worked with just the differentiation of monocytes to immature TC, but also I worked on the maturation and also worked on activation. So even the maturation of dendritic cells is measured by the T cell activation. So if the mature dendritic cells, if the cells attain a mature state, then it has to interact with the native T cell. The native T cell is what I showed as a Th0 cell. If it interacts with the native T cell, two types of T cells are we can get. Okay, I told you one T cell is going to proliferate into two cells. So one type of T cell is a T helper cell, the other is a T helper 2 cell. So whatever T helper cell we got, okay, it releases these kind of interleukins. IL-2, IL-2, IF and gamma, TNF alpha. Okay, the TNF alpha is the major antibody which used for combating the cancer. Not only the cells, the dendritic cells and the T cells, which got an immunity, which got the immune memory, going to combat the particular cancer antigen surface proteins of cancer cells, but also these proteins, tumor necrosis factor alpha, also plays. This protein itself can go and bind with the cancer surface protein of cancer cells, and also it try to inhibit, kill the cancer cells. Okay, everything is possible. Okay, and similarly, the helper T2 cell can also interact with the kind of a B cell, and it can able to proliferate into two types of B cells. One is plasma cell, and the other is memory B cell. The memory B cell has a kind of immune memory, like our B cell memory. Yeah, these memory B cells also try to combat the cancer, and of course, whatever antibodies generated from the plasma cell, numerous antibodies, high quantity of antibodies can have the kind of memory, which that is having a kind of a paratopic region, which can bind with the epitopic region of the cancer surface protein. And the okay, so there are so many mechanisms, not just a single cell, not a single antigen presenting cell like a dendritic cell, but also like a macrophage. Okay, as he had mentioned, B cells, and not only the B cells, the antibodies, the tumor necrosis factor alpha proteins, all these things try effectively to combat the cancer. Okay. And if you see these adjuvants, okay, there are so many kind of adjuvants used in COVID-19 vaccines. If you see the mRNA spike protein, polyethylene glycol is used. And if you see this kind of an ASD1 222 adenoviral kind of adjuvants are used. And for Covishield also, we are using an adenovirus coding spike glycoprotein, which gives the CD4 T cell response, cytotoxic T cell response. And similarly for COVAX, okay, we are using a matrix M adjuvant. So majorly, matrix M adjuvant is composed of saponins, cholesterol, and phospholipid. Did you notify this? Saponins or the plant compounds. So plant compounds can also be used or also used for the preparation of COVID-19 vaccines. And even the efficacy is mentioned, see here parallelly. So for the Sputnik, okay, so adenoviral vectors are used. So not only you can use the plant compounds, you can use viral vectors, you can use other kind of thing like cholesterol, phospholipids. These are other elements for composing an adjuvant. Okay, and if you see this kind of in the next slide, we can see the functional diversity in the adjuvant. So what are the types of adjuvants and what kind of functions, immune functions we can have in our body. So these are mentioned in this slide. Okay, alum, which is the first adjuvant discovered in 1924. or 225. This gives a kind of a sustained delivery at report effect related to T cells. Okay, and nowadays we are getting so many synthetic adjuvants, a combination of many elements like plant compounds, cholesterol. The cholesterol, you know what for they use for delayed release. If you want to get a delayed release of the particular antibodies against the antigen raised, then you can use this kind of a cholesterol. Okay, so this is we are using a kind of a good cholesterol, of course, to improve the efficacy of a vaccine. Okay, in my lab, I have developed one such a software called Adjuvant Immunoanalytical Database. You know, I just developed with nearly 1,000 molecules. So these 1,000 molecules are the products of molecular docking results. We have a software called Discovery Studio. What I did is I docked some plant compounds with 162 receptors of dendritic cells. And whatever doc results I got, I just deposited through the SQL. And of course, foreground, I had a C#.NET framework. And of course, for projecting into the website, I use the personal homepage. Okay, of course, these are also worked by my PG scholars, two different PG scholars. They worked on, they implemented a database in SVC, Steven Glashore Project. Okay, so this is one such achievement we made in our lab. Okay, so this is something about adjuvants. Even we can develop databases if you want for a particular type of a cancer. Okay, I just worked anonymously, like it can be used for any type of cancer, but the size, the data size is just 1,000. We can still able to increase it. We can also able to still increase the interaction so that we'll be getting a precision kind of an information from that. Okay, I have a very small data set. Now let me take you to this B cell epitope prediction. So these are some of the contents. We see each and every slide about these contents. Number one, we'll be discussing about B cell epitope, the classes or the types of B cell epitope. If you see the epitope, generally we have two types of B cell epitope. One is linear and the other is conformational. If you have attended last session, I might have explained these types also. In the linear, you have whatever epitopic peptide you're going to predict is having a continuous. There is no such a gap like this. See the discontinuous? You have a gap here, the length is more. And here, if you see the length is still more, but here the length is less. So why we are getting these kind of discontinuous kind of residues? The reason behind is, if you just take a sequence input, if you take a epitopic sequence like how I taken for T cell epitope on yesterday, you won't find, you find just a linear kind of an epitope predicted from that. But if you instead of relying on the epitopic sequence, if you are feeding an input of 3D structure of epitope, okay, instead of providing a sequence, epitopic sequence as an input, if you are feeding a 3D structure of epitope, obviously, whatever you're going to predict, okay, this have a kind of a conformation, because in antibody you have a heavy chain and a light chain. In the heavy chain and light chain arms, there is one gap. So your amino acid has to go inside. Your epitopic amino acid has to go inside, so that it has a kind of an effective binding. If you tell which is the best epitope when compared to linear and non-linear, which is what is conformational or discontinuous residues, I can tell this is the best epitope. Okay, discontinuous or conform, because it's binding effectively. Okay, so even the residues are flexible accordingly. Okay, so this is something about types of epitope. Okay, so we also see these two types of epitopic prediction on today's session. And we also discussed about the B cell receptor, how it looks like. An antibody, we can find a light chain and a heavy chains. Okay, but we have this kind of you see this variable region only one, but constant region 1, 2, 3, 4. Okay, so you have a constant regions 4, and of course this is also a kind of a complex. B cell receptor looks also like a complex. Okay, mixed with Ig alpha, Ig beta, and these are supported by ITM molecules. And related to this kind of epitopic prediction method, I can take you to an example now. Influenza viral hemagglutinin complex with neutralizing antibody. Okay, there are actually two types of protein molecules in this PDP-ID. One is a surface protein of influenza virus, hemagglutinin, with the antibody which can able to, the whatever antibody here they have shown is going to bind with the viral protein, surface protein, and can kill the virus. Okay, that is the that's what I mentioned as neutralized antibody. Okay, if you say this, okay, hemagglutinin is present in the form of a chain B. Okay, and if you see this, we have chain A and chain B, and whatever antibody, okay, descent from human, okay, is having a paratopic region. This is the paratopic region. I have this protein, whatever I downloaded from the PDB ID, I have visualized using the Discovery Studio visualizer. And then I'm showing you what is a paratopic region and what is the epitopic region. The epitopic region, obviously, this is the region, isn't it? So in this region, okay, do you understand? In this region, you have contact of the human antibody, I mean, the human antibody with the hemagglutinin. So these are the binding sets, of course. But here you don't find any interactions. Okay, here in the chain B, you don't find interaction, but chain is having some kind of interactions. Okay, so now we can go further. So this you can take it as an example to work. Okay, download this. You split this protein molecule separately using the WordPad, and also you can split this protein. You can go for protein-protein docking. There are plenty of protein-protein docking softwares. If you dock it, even you will be getting what are the interacting amino acids of this antibody with the paratopic region with the epitopic region. So for going for, still we didn't get results for the 3D structure of peptide. So we have to wait for some more time. Meanwhile, I can take you to the protein-protein docking softwares. So one such well-known kind of docking tool for interacting protein and protein molecules is patch dock server. If you have two protein molecules, okay, presently I think the server is not active. Okay, if the server is active, then you can try to use this patch dock. Like patch dock, we have say Xdock, SimDock. Okay, there are other softwares also. Okay, Hexdock, which is meant for protein-protein docking. Only thing is that you have to get inside the server and I have to feed two different protein molecules. One might be a major and another might be a minor kind of a macro molecules. So you have to go for the hex doc. Okay, hex doc. Even you can download. Okay, so you can feed your details and you can download the hex doc and you can try to interact a protein with a protein molecule. If you simply type for protein-protein docking, you will be getting plenty of tools. I think now the server is not active. If the server is active, you can also try with the patch dock. So these are some of the tools where you can check the protein-protein interaction. Okay, because even in B cell, we get a kind of an antibodies, isn't it? So one should have a knowledge on that. And now if you see this, another thing, even a kind of a discontinuous linear epitope, the best example is native sperm whale myoglobin. The PDB is mentioned here. So whatever epitope you are going to find, which is complex with the myoglobin, okay, is a kind of a discontinuous linear epitope. I mean, the structure-wise, it's going to totally bind well this kind of a myoglobin protein. Okay, the conformationally, you find a good kind of binding affinity. Now, let me take you to the methods of B cell epitope prediction. Number one is the linear epitope prediction, where here you're going to use the machine learning algorithms. Positive and negative training data sets are used. Combination of one or more amino acid scales are used as an input to one of the machine learning algorithms. You know, BAPPPROACHES, there is one method called BAPPPROACHES. In BAPPPROACHES, you have 1.0 and as well as 2.0. So in BAPPPROACHES, both 1.0 and 2.0, you are going to use a machine learning algorithm called random forest. So using this algorithm, they have developed this tool. This tool is present in, of course, in IEDB. Not only this tool, whatever tools I'm going to show hereafter, including the discontinuous epitope, all these things are available in the IEDB web resource itself. No need to go for other web resources. You remember on yesterday, I was mentioning you for T-cell epitope prediction, don't try to restrict up to the tools present in IEDB, but you can also go for ProPred, MHC, PAN, Net MHC. I told you some of the tools are there. So we have to go to that kind of a server, and also we can predict the epitopes. But whereas for a B cell, this is one of the best website, IEDB, where you have plenty of algorithms, nearly 6 algorithms over there. Okay, see the 6 methods based on 6 different algorithms are there. You can use it for prediction. So if you see the ABC prediction, this is the second tool which uses the artificial neural network for predicting the linear epitopes. And similarly, BC-PRED and FBC-PRED. Okay, this is you're going to use support vector machine learning technique for predicting the linear epitopes. So prediction accuracy is somewhat optimized nowadays. Each and every year they're trying to improvise the algorithm. Okay. Now, if you see the kind of a linear epitope prediction, okay, we have amino acid physicochemical properties are considered to check the binding affinity of epitopes with the paratopic regions. So, what are the major parameters they are focused on the algorithms in each and every methods? If you see, for predicting the beta turns, the Chow and Fastman method has developed a method. Okay, so before going to this, I think I can first show you what are the different tools present in IEDP. Then I can explain you accordingly. So there is one website called iedb.org, which is a free epitope database. This is a large database with epitopes. I mean, for all the types of diseases, can be infectious disease, allergic, autoimmune, cancer. For all these diseases, we can predict the epitopes. So already these epitope informations are fed in the database. Okay. And if you see this B cell epitope prediction, I told you for linear B cell epitopes, you have this hyperlink antigen sequence properties. Click this. And for discontinuous, you have two types of tools. One is Discotope, another is ALIPRO. If you click this linear B cell epitope for the antigen sequence properties, you have few methods. What methods are there for predicting the linear? One is APPPROACHES linear epitope prediction 1.0. If that is not mentioned, this is assumed like 1.0. And this is the latest one, linear epitope prediction 2.0. Chow and Fashman beta turn prediction, Yemeni surface accessibility prediction, Karplus and skulls, flexibility prediction, Polasker and Tongoenker antigenicity, Parker hydrophilicity prediction. So these are the methods. If you say exactly, even if you consider these two as one method, 1, 2, 3, 4, 5, 6, 6 methods are there for linear epitope prediction. Okay, B cell epitope prediction. Now I take you to the presentation. So first, if you see this kind of beta turns, okay, Chow and Fashman has developed a method. Okay, and if you want to work on surface accessibility, Yemeni has developed a method. And flexibility of epitopes, Karplus and skulls has given importance. And antigenic propensity, okay, that is based on the physical-chemical properties of an amino acids. Okay, occurrence of residues in epitopes. Polasker and Tongoenker has proposed an algorithm for it. And based on the hydrophilicity, because even I told you the binding sites are majorly hydrophilic or hydrophobic. Only two types of binding sites are there, isn't it? So if you want to check whether it's hydrophilic or not, so Parker has developed an efficient algorithm for that. Okay, so based on what properties you are going to work on your research project, you can select that particular thing and you can give importance accordingly. Now, if you see this kind of a discontinuous epitope prediction, this is a second method, isn't it? For B cell epitope, you have Discotope and ALIPRO. I also showed you in the homepage of iedb.org. These are the two tools, okay, we can use for predicting the discontinuous B cell epitopes, okay. And what are the properties generally they have used it? If you see, they are using the geometrical properties, not just a single dimension, but they're working on the geometrical. I mean, we are using almost all the 3D kind of an interaction, so isn't it? So geometrical properties, X, Y, Z directions, and amino acid scales used as input to machine learning approaches. Various machine learning approaches are used in this two different methods. Okay, of course, protein-protein docking algorithms are efficiently developed for this discontinuous epitope, B cell epitope predictions. If you see the number one linear epitope prediction method, that is Chow and Fastman, which uses beta turns, isn't it? So how this epitope is predicted actually? This is based on a kind of the occurrence of beta turns. Okay, so actually we have two types of major secondary structures. in proteins. Do you remember? One is alpha helices and the other is beta strands, beta sheets. Okay, alpha helix, beta sheets. So other than alpha helix and beta sheets, you have loops, coils, turns. Okay, you have plenty of interconnecting elements. Okay, coils is of course unique, but you have turns and loops. Okay, but if you see majorly secondary structures of protein, we can tell alpha helix and beta sheets. If you see the poor formers of alpha helix or proline and glycine. If you have studied the protein science or the structure subjects, you might have come across these two amino acids, where you can also find these two amino acids to determine even the stereochemical quality of a protein. If you are predicting the 3D structure of a protein where the structure is not known, and if you have the sequence alone, so after the prediction of the 3D structure, you are going to use Ramachandran plot to assess the structure, isn't it? So that they have given importance for these two residues, proline and glycine. Okay, so these are of course the poor formers of alpha helix. If these amino acids are not forming the alpha helix, then where you can find this kind of residues? You may find in loops, turns, or also sometimes in the beta sheets. The turns, especially we can find in turns, isn't it? So based on the propensity, okay, based on the propensity to form alpha helix and beta sheets, okay, you can able to find, you see the 20 amino acids are shown below in the scale, okay, which is having, if you see the glycine G is having a high value 1.56. If you see the proline, it's also having a high value 1.52. Okay, so the Job-Fastman method takes into account only the quality that each individual amino acid will appear in alpha helix. You know, proline and glycine, due to the unique conformational properties of the peptide bonds, commonly end a helix. So you will be going for the lowest value amino acids for predicting the beta terms. Okay. Now we can go for AIML surface accessibility scale. Here, if you see the AIML surface accessibility scale, the accessibility scale was calculated using this formula. You know, here the surface accessibility, like if you have a kind of a hydrophobic coach binding site or a hydrophilic binding site, then only you can able to find even your epitope can bind efficiently with a paratopic region, isn't it? So it's not like a small molecule. Whatever epitope you predict is a 9 amino acid or 13 amino acid. It's a macromolecule. It's a polypeptide, of course, a small polypeptide. If an entire epitope has to go and bind with the paratopic region, then it should have a proper conformational accessibility, conformational flexibility, and even the surface accessibility. The surface accessibility of interaction of epitope with the paratopic can also be fine with this. Okay, so hexapeptide sequence is SN greater than 1 engaged. So if value is more, then you have more binding regions. If you see R is having 0.95, K lysine is 0.97. These are the most important binding sites, of course. Okay, if you have the K and R in epitopic regions, these can bind efficiently with the paratope region of antibody. So this is the meaning of AIML surface accessibility. And now we will be discussing about the flexibility. Even the amino acids present in the epitopic peptide should be flexible, isn't it? If and if it is flexible, then only it can bind effectively with the paratopic region. So to measure this, corpus and skulls has measured the scale for the amino acids. And Kolaskar has developed a scale for antigenicity. For antigenicity, there are few parameters, few physical and chemical parameters of amino acids. So these are determined by Kolaskar and Bogankar. And now, if you see in this slide, this is about B cell epitope prediction softwares. So not only we have softwares in IEDB, but we also have private. We have other kind of web resources to work on B cell epitope prediction. What are that? So if you see, other than Elitope and Discotope, we have CEP, we have Pepito, we have SEPA, Epitopia, EPSVR, EPIPRED, PEAS, Memox. Okay, so we have Pepito. There are so many kind of conformational B cell epitope tools available in internet. Like even for conformation, even for linear, you have these kind of tools. Okay. So other than IATP, see, there are so many tools developed from the Indian scientists, IMTech, popularly meant for purchasing the cell lines, the cells, microbial cells, are also predicted this kind of a B cell epitope prediction tools. Okay, the website developed by Raghava scientist. So if you see this B cell epitope prediction tools, we have these options for prediction of linear as well as the discontinuous epitopes. Okay, for going for modeling and docking of antigen the protein molecules, also we have a kind of hyperlink. You can click to that and you can try to um, complete your job. Simply go to the website, just feed the input sequence. To remember what sequences I used as an input, I have taken the W protein of Nipah virus last session. The same sequence, you feed it as an input and click any of the methods and click submit, you get a kind of result. How you will be getting? So even you can not only can paste the sequence, if you know the UniProt ID of the specific protein, just type it. Either paste a fast protein sequence. Here they mentioned not to paste a FASTA format, but a protein sequence alone. It means that delete the first line. You just paste the amino acid sequence alone here, or just paste the UniProt ID. And no, sorry, what happened? Yeah, I just take you to this page. So UniProt ID or the amino acid sequence, click submit. Now go to the APPPROACHES. In APPPROACHES linear epitope prediction, okay, after pasting this Nipah virus W protein, you got so many peaks. You got so many peaks actually. Whatever mentioned in yellow color, okay, or the best kind of an epitopes. Okay, when compared to the green is not is actually we have a threshold. We fixed a threshold to get the best epitopes. So I fixed a threshold like 0.5. So I got numerous epitopes. But if I want to get only the best 10 epitopes, then you can see you can fix the threshold as 0.7. If you see in the 0.7, we have one epitope region and second epitope, three epitopes. So we got nearly three best epitopes in the threshold 0.7. The taller the peak, the best epitope you are predicted from the APPPROACHES algorithm. Okay, so this is how we can able to. And if you see the results, not only you can get in the form of a graph, but also you will be getting in the form of a tabular form. In the tabular form, if you see here, in the 20th position, okay, only from the 20th position onwards, you find the epitopic region, isn't it? So here also you can find the same. The 1 to 19, you don't find any epitopes because you got a kind of a green color which is below the 0.5 threshold. So you don't find any epitopic regions here. But from here onwards, you find epitope regions. So whatever amino acids you have from the 20th position, okay, you not only you are getting the score, but you're also getting the assignment as E. E stands for epitopic region. Okay, so this is an amino acid and this is the position. Now, of course, the score, the maximum the score, it is a more epitopic type. Okay, so this we predicted from APPPROACHES. Similarly, even in Discotope, instead of simply pasting the sequence of a W protein, you should have a kind of a 3D structure of even the epitopic peptide. Then only you can able to dock and you can check it using the Discotope. Okay, so what are the inputs generally you need? You need a PDB ID. You need a PDB ID. Okay, so first you should have a PDB ID of the 3D structure of a protein. You just upload it and you just click submit. So after this, if you have a 3D structure of W protein, okay, if you submit it, no. Where is the Discotope result? So if you submit it, you will be getting a result. Now I am showing you Chowan-Fastman beta tone prediction results. If I get a 3D structure of an epitope, I can show you that. Still it's running. So for the Chowan-Fastman, similarly, however I explained for the earlier APPPROACHES. Okay, here also in Chowan-Fastman, the maximum peak gives you the best epitopic region. Okay, and similarly even for the MNE surface accessibility, the maximum gives the best epitope region. So I can even in Parker hydrophilicity, I have taken this as the best. because you got a maximum peak here. But corpus and skulls are shown in these regions. If you see, these peak regions are not matching with each other. Maybe the hydrophilicity is based in this epitopic region. But if you see the flexibility, these are the best regions. The same amino acid positions, but the peak wise, you can find a different kind of a positions. Okay, but you have to use the machine learning algorithms to find the best peptide from that. This might be one peptide, this is second peptide, this is third peptide, this is fourth and fifth. If one or two are matching with the earlier algorithms, then you can choose from these two algorithms which is matching with the high peptide region. So I can take this as a best epitopic peptide. Okay, you have to carefully select the best epitopic peptide. You can't tell this for 10 best epitopic peptides can be used for treatment. You have to screen it to as many as minimum number of epitopes. Okay. Of course, we can use a multi-tope vaccine. You can also design this epitope and use the second epitope and third epitope and fourth and fifth. You can mix all these 5 epitopes in a single multi-tope, and even that can be used for curing. But if you find any coherence between this kind of an epitopic region, you can reduce the number, and so that it will be easier for developing the product. Most of the Chinese companies are involved in developing this kind of an epitopic peptides. If you just design and if you just send it to the Chinese company, they can send it to you. Okay, nowadays even Indian companies are coming forward to design the peptides for cancer as well as for the infectious viral or bacterial diseases. Now, whatever we are designing, okay, it can be a 3D structure of a peptide, okay, B-cell epitopic peptide, especially for discontinuous residues. What will be doing with that? We have to dock with a receptor, isn't it? So if you see the T-cell epitope, the efficacy of T-cell epitope is measured by docking with MHC molecules. You have to model the MHC molecules from the HLA genes, and then you try to dock your T-cell epitopes with the MHC molecules. But how about B cells? The B cells has to dock with toll-like receptors. This is most important for innate immunity. If the foreign antigens are entering into our body, then obviously the toll-like receptors will try to give an innate immunity in the beginning, isn't it? So if you are focusing on a bacterial kind of invading pathogens, then you have to target toll-like receptor 4. I just focused on Nipah virus, isn't it? It's not a bacteria. So instead of going for TLR4, I can use TLR7 and 9. So these are, of course, major receptors which combat the viral viruses. To combat the viruses, these are the major toll-like receptors, TLR7 and 9. Similarly, for different, for cancer and for other kind of thing, we can use the types of toll-like receptors. So you should have a 3D structure of toll-like receptors, and you should have a 3D structure of your epitopic peptide. Then dock this 3D structure with this 3D structure of the toll-like receptors. This is how we can able to check the efficacy of a B-cell epitope, whatever you have predicted. Finally, you have to do this step. Okay, and this is the final slide from my presentation. This is a FDA-approved cell and gene therapies. If you see for oncologic, for a cancer kind of cell therapies, I told you that for the cancer treatment, not only can use the epitopes, but we can use the entire cells. We can use mRNA. We can use we can use other molecules. Okay, from the major sources. Okay, and we can able to treat, isn't it? So if you see the major in the world, okay, the Adcitel, Dendrion, Provenge, BioX. So these are some of the companies majorly or for providing the therapies, both cell and gene based for treatment of cancer. So this is something about this statistics is taken in 2023 throughout worldwide. So these are the major companies applying for products for the cancer. Okay, now I was explaining you something about antibody prediction and taking you to the 3D structure of PEPFOLD3, isn't it? Still, I am not getting the structure for PEPFOLD3. I think we have to wait for some more time. I can check whether I have any kind of a results already predicted from WebFold 3. If I have, I can show you that. WebFold 3, WebFold server. If it is not available, I do one thing. I can download it out today, and tomorrow I can show you. Okay. So only thing is that you have to go for having a 3D structure of this and that for talking. Now I just take you to this kind of linear epitopic prediction. Only thing is that you should need uh, a UniProt, a SwissProt ID or UniProt ID, or just a protein sequence. In my PPT, I mentioned the name of UniProt ID. You remember what is the UniProt ID I fed? If you see the UniProt ID, I mentioned as P0C1C7. P0C1C7. I think this is the UniProt ID. Simply, I'm going for APPPROACHES linear epitopic prediction submit. So for that particular Nipah virus W protein, I'm going to predict epitopic regions. Like however you predicted for T cell epitope, here also will be the similar kind of procedures are happening. Okay. You get an output like however I shown in the PowerPoint. You can also go for the other prediction methods. I don't know. You're getting some kind of security. Is that the same? P0C1C7. P0C1C7. So even the residues maximum should be this, and minimum 250 residues, 50,000 residues. I go for Chow Fastman. Provide an example and get an output. Yeah, Chow Fastman are getting a result. So these are the top peaks. If you see this peak, this peak region is in the 140th, 142 to 150, something like that. Okay. So if you see this 142 to 150, so 142. So here you can see the score is increased tremendously. 1.413, 1.327. These also best regions. So if you see this, I can tell if you want a 9 amino acid peptide. So based on the scores, if you take, okay, this will be the best 9 amino acid. This will be the best 9 amino acid. Okay, this is how we can select based on the scores. Yeah, any doubts you have? Participants are almost completed how to predict the B cell epitopes present just because you get a 3D structure. I just explained you how to go for a linear B cell epitope prediction. One such method I've shown for Tau Fastman. I can also try for the other methods. I have completed my session. Now, if you have any doubts, you can ask me. If not, thank you very much for the TLR tool like receptors. Tool like receptors for innate immunity. These protein receptors are meant for innate immunity. Okay. I just stop presenting. Any other doubts you have, participants, you can ask. If no doubts, again the same. Thank you very much for